Hi, good morning, everyone. Thank you very much for attending the Gene Talk. I'm Kevin from the Memorial Children's Hospital in Delaware, and also research scientist and lab head in the PETCHEM Center. So today I'm going to talk about non-invasive detection of neurofibromatosis. Later on, we call it NF1 with positron emission PET tomography imaging. Oh, it looks like sorry. Yeah, first I'd like to give you a very brief background information. So my research mainly focuses on molecular imaging. So molecular imaging is a type of medical imaging that provides valuable information to what happens inside the body at a molecular or cellular level. So here the picture, let's take the cancer for example. We inject the radioactive imaging agent to the patient. Then the imaging agent home into the tumor region to improve the contrast and wash out from the lung target region very fast. At the same time, when we label the compound with different isotope, that imaging agent also can be used for therapy. So our ultimate goal is the imaging agent to aid diagnosis, to monitor disease progression and respond to treatment. At the same time, in the future, we're also interested in the different radio isotope to use for the cancer therapy, use different radioisotopes. So this slide shows the basic concept. What we are doing, we involve the radioactive imaging from the bench to bedside clinical translation. The first, we typically work with the physician clinician to identify the clinical challenges. Then next, we identify the target we are interested in. So after that, we identify the target towards a specific disease. So the next step, we design and synthesize the new compound. The new compound typically have high binding affinity and high selectivity toward the target. And one major part of our research interest is focused on neuroimaging. Let's say, for example, for neuroimaging, when we design the new compound, typically the new compound have the high binding affinity less than 10 nanomolar toward the receptor or enzyme. So after we identify the promising compound, we radio label the compound with appropriate radioisotope. For example, it's copper 11, fluorine 18, and gallium 68 or copper 64. After get the chemistry and radiochemistry down, we start evaluate a radiotracer in the cell level and animal models in terms of biodistribution study and PET scans and blocking study or the radiograph study and the metabolic analysis to look at the whole body distribution or connected of the new tracers. If from all the preclinical study in terms of cell level and animal model, the tracers are still very promising, then we start a clinical translation of the tracers. So to address the clinical challenge, one key part is to identify the biomarker, the target. Just like Dr. Aikens yesterday gave us an excellent talk regarding the DNA methylation is the important biomarker in CP. So typically the different disease involve different biological activities. So we use a different tracer to look at the different biological activities, such as use FDOPA to look at the ligand receptor, to look at dopamine D2 receptor, and also use FDG to look at the metabolism. FDG most often use the PET tracer for the look at the tumor metabolism. Also use a short half-life O15 to detect the angiogenesis in the brain. Also use F18 level F-mesol to look at the hypoxia of tumor, because we know that the tumor environment typically there's a relatively surrounding environment that's a low content, very oxygen, and also the pH environment also very different from the tumors, and also use FLT to look at the tumor proliferation. So here are just a very typical examples to look at the biological activities in the different lesions, and also to use a typical ray tracer to look at the biological activity, address the challenges. So this cartoon will show the typical design of radio tracers. Sometimes we also call the radiopharmaceuticals. So typically radio tracer composed of three parts. One is the radioisotope, with radio label with compound radioisotope is called radioactive compound. Another part is target molecular. Typically, the target molecule has high binding affinity, just as we mentioned before, and because the cancer cell surface is highly expressed different protein and receptors. So another part is the target is called a radioligand, or target molecule have high binding affinity towards the surface of cancer cells. So we use a linker connect between the radioactive compound and the target molecule. That's the three major components called the radiopharmaceutical that target cancer cells or other disease patterns. So this picture shows the FDA-approved tracers. So around 20 PET tracers approved by the FDA, and we can see that most of our tracers are used for the tumor and the neurodegenerative diseases. And particularly, we can see that in 2020, there are 4 PET tracers approved by FDA, and FDG, that's the most common used PET tracer in both pediatric population and adult populations. So in our NEMOS PET Center, we involve both clinical investigation and preclinical research. And here shows a typical example of what we are doing in the clinical PET Center. We use a tracer called G180 to look at the neuroinflammation. And for the MRI, for the FLAIR MRI, we can see that there are multiple lesions for the MRI in the second picture and the second picture. However, for the functional PET imaging, we can see that only 2 active lesions exist. So that means that regarding the functional PET tracer is a very powerful tool to look at functional changes compared to the MRI, and they can differentiate the active lesion and the versus inactive lesions. That's a major function of PET, to look at functional changes or biomarkers, because we can see that the MRI, there's multiple lesions, but some is active, some is inactive. Maybe the when we look at the history, the patient takes some drugs to anti-inflammation. So that's why we only found inactive lesion we found it in the patient. So another key part of the preclinical study, we use a tryptophan tracer to look at the tryptophan metabolism. So this slide shows the basic background information. So the tryptophan is an essential amino acid. So tryptophan cannot be synthesized by mammals. Tryptophan must be obtained from external source. So in our human body, a majority part of tryptophan is metabolized through the clearing pathway. The clearing pathway is catalyzed by 2 key enzymes. It's called ADO and TDO. So under normal physiological conditions, the tryptophan downstream metabolite is called quininic acid, can convert to NAD. NAD is the important coenzyme utilized by all living cells. However, under pathological conditions, excessive accumulation of quininic acid can cause neurotoxic effect and inhibit neuroprotective T cell function. That's the cause of the many diseases such as epilepsy, neuro-oncology, and Parkinson's disease. So imaging of clearing pathway can provide valuable information to provide non-invasive cancer immunotherapy response. So here shows how we synthesize the radiotracer in the GMP facility by one-part, two-step strategy. So here, and the bottom picture shows the control panel and the diagram of the module to develop the tracer in the clean room. So early time, we collaborate with Xuyi, use this tracer to look at the medulloblastoma in the animal model. So medulloblastoma is the most malignant brain tumor in pediatric population. So first we use the tracer to look at the static scan in the brain tumors. So in the top left, we can see that this tracer has high uptake in the brain tumors and low uptake in the control tumor. And the static scan in the right side, a 3-hour dynamic scan, we can see that the tracer has high uptake in the brain region and the low uptake in the contralateral and the referral region. Then we also compare the tracer with FDG, just as we mentioned. FDG is the most common used PET tracer in the clinic. And in the bottom left, we can see that the tryptophan tracer high uptake in the brain tumor and a low uptake in the background. However, for the FDG, commercial available FDG, we can see that the tracer high uptake in the target region. However, at the same time, we can see that FDG also has global long-specific uptake in the brain region. We also compare the imaging, because the Tryptophan tracer, we know that the amino acid have two configurations, L and D. We also compare the tracer uptake between the L configuration and D configuration, and we can see that at different time points, the L amino acid have 5 times higher uptake than the D isomer. So that means that the amino acid enter the brain tumor through the L amino acid transporter. So to address the clinical challenge when we talk to physicians, there's another clinical challenge is regarding neurofibromatosis type 1, because neurofibromatosis 1, most of the time is a benign tumor, is a complex autosomal dominant genetic disorder that have an incidence in 1 per 3,000 children. And most the children with NF1 that a benign tumor, but there's more or less have the cognitive dysfunctions, and also 50% have ADHD. But anyway, most of the patients, the tumor is benign, but for the clinical side, about 8 to 16% of patients develop malignant tumors. The malignant tumor have a very high mortality rate with a 5-year survival rate of 30%. And the clinical side, with MRI or CT, we can detect the lesion, but sometimes we cannot tell the lesion is malignant or benign. And the same for the FDG. FDG maybe can look at the hot spot, but sometimes we cannot monitor the progression and cannot tell the tumor is benign or malignant. So we hypothesize that our Tryptophan tracer may detect the NF1 and also have a chance to look at benign tumor, differentiate the malignant tumor. So our hypothesis is mainly based on this paper. In this paper, they studied a lot of human NF1 samples. They found that in those human NF1 samples, the Aryl hydrocarbon receptor is AHR receptor highly expressed in the NF1 samples. At the same time, and study showed that the Tryptophan downstream metabolite is the endogenous ligand for AHR. So the highly expressed AHR and in turn contribute to the high expression of TDO. And we know that TDO is the key enzyme for Tryptophan metabolism. So we hypothesize that in the NF1 samples, the Tryptophan metabolism rate is upregulated, and the upregulated Tryptophan metabolism rate is correlated with the high TDO expression, and also contribute to the high uptake of Tryptophan tracer, which is in turn correlate with the PET imaging results, also correlate with the malignancy. That's how the major hypothesis in this project. So first, we studied the tracer, the Tryptophan tracer FDG on the same mice, the healthy control mice. And here's the picture shows that we can see that the Tryptophan tracer have very low uptake in the brain, but for the FDG, that the long tracer, we know that have global long-specific and brain uptake, because we know that the brain consumes about 25% of energy. So FDG, no matter the animal study and human, we can see that the FDG have global brain uptake. The next day, we do the genotyping to identify the mice have high chance to develop NF1 tumors. And for this mice, the commercial available mice, and the mice have triple mutations, TRP53, SUZ112, and NF1, 3 mutations have high chance to develop tumors. After we identify the mice with have high chance to develop tumors, and we monitor the mice with the Tryptophan tracer. And here shows the mice 1 and the typical example of mice 1 and mice 2. We can see that the Tryptophan tracer have high uptake in the F1 tumor. And one major difference between this animal model from the medulloblastoma we previously talked about, we can know that the medulloblastoma is developed in the cerebellum of the brain. But for this animal model, it's transgenic animal model. We cannot predict where the tumor, when the tumor grow. So we can see that here the mouse 1 is the tumor in the limb position, around the neck position, but right side in the mouse 2, the tumor in the spinal cord position. And the picture, we can see that MOS2 also on the right side, it also shows the tumor around the spinal cord. But for both the neck tumor MOS1 and the spinal cord MOS2, we can see that this tracer, we can clearly look at the tumor uptake of the radiotracer. So we also, let's say we do the biodistribution study of this tracer in the wild-type mice and the trivial mutation mice. We can see that the pancreas have the highest uptake, followed by the heart and liver and spleen. We can see that the brain uptake was low in the wild-type mice and trivial mutation mice. So that is consistent with what we observe in the PET imaging. And very important, we can see that the bone uptake was low and stable during the 90 minutes biodistribution study. That's very important because many tracers cannot move to clinical trial because it's not stable in vivo if differentiation. And if differentiation, we know that free fluoride have high uptake in the bone. If high uptake in the bone, that means that the tracer is not stable and probably will confound imaging analysis. So the image, particularly for F18 level tracer, is stable towards differentiation is very important. So from the tumor mice, we can see that the different tracer have high target to long-term ratio, particularly with the tumor to fat ratio, which about 6 at 90 minutes post-injection tracer. So we just present result in the SNMI meeting in the Vancouver in Canada. So we just show the biodistribution study and the static PET imaging. We also do the dynamic scan of the mice, and we can see that the tracer in the tumor region increased slowly and peak at around 30 minutes post-injection. And in lung tumor region, we can also see some uptake, but the target to non-target ratio was high at 30 minutes. And this picture shows another tumor mice. We can see that around the bottom of the spinal cord, and also we show the tracer uptake in the spinal cord. So we also do some staining to compare the health control tissue and with the lesion tissue. We can see that the significant difference with the morphological changes between the healthy tissue and the lesion tissue. So we are actually working to look at ideal expression of the lesion region to correlate with the imaging result. So here's the conclusion of our study. The first, we radiosynthesize the Tryptophan tracer in GMP facility with high radiochemical yield and high enantiomeric excess value. The biodistribution study showed that the pancreas had highest uptake, followed by the kidney liver. And one important result, we can see that this tracer is very stable towards differentiation. That means that in vivo, we can have high potential to use the tracer to other disease model or move to clinical translation. In fact, we submit the IND, EIND to FDA. This tracer approved by the FDA for first-in-human investigations. And also, we can see that both dynamic scan and PET scan result, we can see that at different different tumor locations, we can see that this tracer have high uptake in the tumor region. And compare FDG, particularly for the brain uptake, we can see that the Tryptophan tracer have a very low uptake in the brain, but the FDG, you can see that have global uptake. That means that this tracer may be very promising for brain tumors, neurological and other neurological conditions. So last, I'd like to thank all of the collaborators, particularly Sigrid Rahul, on this project. I also like to thank the radiation safety officer from the clinical side and the preclinical side. And also like to thank the histology, chemistry, and the tissue processing core regarding the HE staining. And also I appreciate the Socrates from Delaware State University regarding the image analysis. So this project was supported by the DCTR pilot grant and the Department of Radiology and the NEMOS by the Biomedical Research. Thank you.